So welcome to the 2021 Symposium on Neurotechnology. We have this annually, and this is our first virtual one. And we've had this program a few years now. Started back in, what is it, Dan, 2015, when Dan and Nader and I started talking about this kind of program, and very excited to say that we are now officially funded by the NIH to have a T32 program starting this year. So I'll give you a little bit of background on this program here. All right, so I wanted to start with a little background on the Center for Neurotechnology, where our aim is to bring together clinicians, engineers, neuroscientists, so that we can, accelerate translational science. And the directors, we are very happy to welcome you to our annual symposium. We have 3 directors here because this is a very interdisciplinary program and really aiming to bring together neuroscience, engineering, and translational science. And so we have myself and Dan Markovic and Daniel Liu to oversee the program, which includes the training program for the T32 program, as well as the weekly seminars, as well as this annual symposium and other activities that I'll explain about. So UCLA, when we were in person, of course, really is lucky to have these 3 very interdisciplinary fields in close proximity. So I can't wait until we're back on campus and I can walk down across the way to Dan's lab in engineering. You could see here the red is where the medical school is, where it houses a lot of the neuroscience labs and the neuroscience buildings. And then we have yellow. Yellow is actually some additional neuroscience buildings outside of the medical school. So a lot of neuroscience labs here at UCLA, very fortunate. And then engineering, which is just, a building away, houses the various programs such as electrical and computer engineering, neuroengineering within the bioengineering program, and others. And our goal here is to really bring together these fields together with regulatory and contracting to facilitate technology transition in the brain sciences fields. Our mission is to increase the efficacy, decrease the burden, and accelerate implementation of neurotechnology for medical applications. And as you can imagine, this spans a very wide range of technologies. And so you're just seeing today some of those technologies that are coming out of UCLA, but there are many, many more that we're not able to fit in, and we hope to fit them in throughout the subsequent years. There's a lot of investment and funding coming into this area as well, which we're very fortunate and hoping to take advantage of this time to really accelerate these technologies. So we are, like I said, now funded by the NIH. Our T32 program funds pre- and postdoctoral fellows. We have 4 funded and 4 unfunded positions each year, and the funded positions and the unfunded can be kept for 2 years for the trainee, up to 2 years for each trainee. And this is, again, spanning multiple departments at UCLA in the engineering, neuroscience, medical school, as well as the Ph.D. programs across campus. So here are our fundees, in case you haven't been introduced to them for 2020. So congratulations to those of you who are here and are presenting today. We have 6 predoctoral and 2 postdoctoral fellows, and we're very excited to welcome them. We've had weekly seminars with them so far, and really, have adapted to this new virtual teaching environment. So our objectives are, again, to understand the spectrum of translational neurotechnology research, and hopefully the trainees can gain expertise not only in their specific projects, but also in terms of the process of clinical translation. So really taking the bench to bedside approach literally here, and learning about the whole process, learning how to define clinical needs. So as engineers, really understanding the clinical needs for a particular technology in order to accelerate the translational aspect of that technology. The trainees are mentored by a statistician and also take courses in our weekly seminar on statistical methodologies. So they're also trained in scientific rigor. We also have seminars coming up in the second year of the program on regulatory pathways and business of science, including, of course, communication. And the trainees will get exposure to the industry side and understanding of industry perspectives and regulatory and business hurdles in translational neurotechnology. So it's really, like I said, from nuts, from bench to bedside, all the way spanning the entire progression of neurotechnology development, testing, and the research that's involved, as well as translation to the commercial side. So you can learn more at our website here, and we have weekly seminar series, and this is open to not only the trainees, but other students and members of the community, where we have 80 or so plus seminars that include external faculty presentations, internal faculty presentations, presentations from industry. We have our regulatory and business lectures, our statistical lectures, and also career development in terms of communication and various other aspects of career development. Here today we have our annual Symposium, of course, and we'll have this every year. Hopefully next year this will be in person. And then also another aspect of the program is each trainee gets triple mentorship. So they have mentorship from an engineer, as well as a clinician, as well as a statistician. And then, of course, within that, many of these faculty members are also in the neuroscience field. So they have sort of a neuroscience, engineering, clinician, statistics mentorship. So it's really covering all aspects here. And the goal is to really train these trained trainees in multiple aspects of this field. Clinical immersion, we have in our program where each trainee is paired with, again, a clinical mentor, and they will be able to see aspects of the clinical setting, clinical treatment, and really understand the clinical needs. COVID, really put a little bit of a, made this a little bit more difficult this last year, but UCLA is in good shape now and reopening, and so we hope to resume these activities. And we have, of course, other topics that are included in the training, and including a submission of individual funding applications and others. So feel free to email myself or the TNT email address if you have any questions on this program. And then pre and postdoctoral scholars are eligible for this program. We have, dozens of faculty members that serve as mentors within this program. And like I said before, up to 2 years, trainees can participate. All right, so that I think is all I have for the introduction here. Happy to take any questions you may have on the program before we get started and give access to our next presenter while we do that here. Thanks, Joe. So today we have 3 sessions. We broke it up in terms of the different aspects of the program. We're starting out with more neuroscience, neural dynamics, and computational modeling. And then we'll go into technology and clinical applications. So Gina Poe is a professor in integrative biology and physiology, and we'll talk today about precise techniques for measuring and controlling sleep circuit features to improve cognition.
Good morning, everyone. I'm so excited to be part of this first Symposium after we've gotten our training grant. And I want to thank the organizers for all of the hard work they put into this. I'm glad to be here to represent my laboratory. This is us at during the pandemic. And I'm going to be talking a little bit about the work of Raquel Guthrie, who will be presenting a little later today, and Michelle Fraser. Both of them are graduate students in my laboratory. And just mention here Karina Kuss, who will be starting graduate school, a PhD in neuroscience here at UCLA in the fall. And I just want to shout out the great work she's been doing on developing a really cool sleep scoring program that would be useful not just for us, but for the whole world and all species, hopefully, or at least mammalian species. All right, so this is how our sleep changes across our lifespan. And you can see when we're born, we've got a lot of sleep, a lot of REM sleep, and this N2 stage of sleep. the amount of slow wave sleep doesn't change that much until we get very much older, and then it reduces quite a bit. So let's see. Yes. So so one of the things that changes with sleep is changes also across our lifespan is our ability to learn and put things together. As you know, when we're very, very young, we can learn, we're like sponges. We learn things really, really quickly. And as we get older, that becomes a little more difficult. Our brains become perhaps a little more full, you might want to say, of things that we already know. It's a little more difficult to learn. Of course, this varies from person to person. One of the things that does correspond with our ability to learn is the number of slow waves also. So this is just the various functions of sleep. Slow wave sleep has recently been found to clear the garbage out of our brains. So as we get older, actually the amplitude of these slow waves gets smaller, and this garbage gets cleared through the lymphatic system. We're also restoring energy from adenosine to ATP during this state. This is what caffeine blocks, is the receptors that show that adenosine is built up. And so we don't feel as sleepy, but adenosine is a powerful sleep-inducing signal. And then we build our proteins during this state in large quantities. So this is an important state for a lot of things that are involved in brain health. And during the transition to REM, which is that N2 state I showed you in that last slide, we're also integrating and consolidating information. And we do that through something called sleep spindles, which are about 1.5 seconds long. And the density of our sleep spindles correlates with IQ. This is a time when our hippocampus, our memory structure, is really coordinated with our cortex to transfer information and consolidate information that we've learned during the day to the rest of the brain. And then during REM sleep, my research has been showing that it's really a time to clean unnecessary synapses, to build and connect new things that we might not connect during the daytime. And we do that through the theta activity that happens in the hippocampus and coordination between the hippocampus and other areas. We're weakening old connections and strengthening new ones in order to sort of come up with new insights, for example. Okay, so there are lots of people around the world right now who are trying to pump up our sleep amounts to counteract the effects of aging or dementia, or they're trying to amplify particular sleep features like sleep spindles, which are lost in people with schizophrenia, or slow waves, or which are lost or changed in Alzheimer's disease. But I'm going to point out today why this strategy, this kind of naive strategy, won't work. Here's why. And that's because it's not just the amounts of these things, but the timing of the coincident features that happen naturally during sleep that is really important. Timing is everything. That's one thing to think about. And the other thing is the place of activity is important. Location, just like the real estate agents say, in our brain, various things are happening in different places. So just a kind of a global amplification of various features or across the whole brain is just not going to work. So here's some early data that I generated when I was a postdoc. It was shown already that long-term potentiation, the building block for learning, really occurs when you stimulate specifically at the peaks of what's called a theta rhythm, which is that thing you see during REM sleep and also during waking. That's a 5 to 10 hertz rhythm. And if you stimulate at the peaks of theta, you get beautiful long-term potentiation. But if you stimulate the troughs of theta, which is just 100 milliseconds later, you will get actually theta trough activity. I mean, you will get depotentiation, the opposite of long-term potentiation. Both are important for learning and memory, but the timing of activity in relation to this is really important. And what I found is that during waking, activity in relation to theta is bursting at theta peaks, consistent with building memories. But during REM sleep, activity is occurring at theta troughs, which is consistent with cleaning those synapses that have already been consolidated to the cortex. And this happens with memories that have been well consolidated already to the cortex. So it doesn't happen until that consolidation has occurred over the period of about a week. So, so this is just in general a kind of gestalt of what I just showed you, which is that novel memories are consolidated and reactivated during REM sleep at theta peaks as opposed to theta troughs. But if some memory is already familiar, then the firing switches to theta troughs rather than theta peaks. All right. So there was a really cool paper just 2 years ago in the summer of 2019 that showed also that there's subtle differences between types of activity. So there's something called delta activity, which is 0 to 4 hertz or something in the something like that, 0 to 4 hertz in the brain of all animals, all mammals that have been measured. And what they showed with a neuroprosthetic device learning paradigm in rats is that if they inhibited activity during the slowest section of that slow wave, during slow oscillations, reactivation that normally occurs, if they used optogenetic devices to inhibit activity at the peaks of those slow oscillations, they got a huge reduction in the ability of animals to learn that neuroprosthetic learning technique. But if they instead inhibited activity in the cortex at the peaks of delta, which is just a little bit faster and a little bit more shallow and a little bit more local, then they got actually an improvement of activity in these animals. And the reason why they think they got this improvement is if they inhibited activity during delta, they actually drove sleep spindles, those 1.5 second long blue things I showed you a couple slides back, they drove them into the arms of those slow oscillations. So they amplified the slow oscillation activity. So it's not just amplifying the amount of slow wave sleep, but you really need to figure out which waves you want to amplify versus inhibit in slow wave sleep if you want to improve cognition. One last thing I want to show you in terms of timing is the locus coeruleus, which projects all over the forebrain and helps us quickly learn and have LTP. That is also really precisely timed in relation to these slow oscillations. So here's the way that the locus coeruleus normally fires just before the peaks of this slow oscillation. And that's where memories are reactivated. So if you are cause the locus coeruleus to misfire in relation to this timing, you're going to actually cause actually a reduction in sleep spindles. This is something we did. And also perhaps not provide norepinephrine at the peaks of the slow oscillations when all this reactivation is happening. So you're not able to help the brain learn quickly from all that reactivation that's happening in slow wave sleep. And one of the things that happens with Alzheimer's disease is the locus coeruleus is the first to degenerate from a buildup of plaques, and the firing of the locus coeruleus gets affected. First, it becomes hyperactive and then hypoactive. We optogenetically stimulated the locus coeruleus across sleep, made it hyperactive across sleep, and we didn't wake up the animal, but we were able to reduce these numbers of sleep spindles that happen that are normally nested with these slow oscillations. And that really correlated with the ability of animals to learn this task. All right, so Michelle Frazier in my laboratory is really doing some great work. She is recording the LFP of mice and the EMG nuchal signals in order to be able to detect. She's sending the signals to OpenEFES and using a Python plugin, a program that she wrote to do real-time detection of REM sleep. And she's using delta theta ratios and EMG activity. And when she's able to detect REM sleep immediately when it occurs, those theta waves, she's able to send activity to an Arduino pin that then turns on an LED driver to give optogenetic stimulation or inhibition of specific areas and specific types of cells, which has been working really, really well. It's a closed loop system. And so given the precision of timing of these various sleep events that I just told you about, the precise timing of this stimulation and inhibition is really important. The amounts of sleep overall are not independently important. The amplitude of waves, not necessarily independently important. But really it's, and that's been shown because a lot of these colleagues around the world who are trying to do sleep dependent stimulation to drive the amplitude or occurrence of sleep spindles or sleep slow waves are really coming up with a mixed bag of effects that they can't really explain. And that's because I think this timing in relation to sleep events is important. And then the next point I want to talk to you about is the sleep features vary between brain structures. So we really need to measure where you want to record and where you want to amplify these sleep events in various parts of the brain and not just all over at the same time. So this is a study that we actually published in 2012. I'm sorry, this is an old thing, but what we showed is that in the hippocampus versus in the cortex, you can have REM sleep here appear for long periods of time. This is 20 seconds at a time when the cortex is still in slow wave sleep. And this dissociated state, when the hippocampus is in the theta wave reactivation state and the cortex is still in that sleep spindle state, is really an important time for consolidating information to the cortex. We found that this appearance of dissociated states where there's more REM sleep in the hippocampus than the cortex appears even more strongly when an animal is really trying to learn and able to benefit from sleep. To their learning is benefited. And then the last point I want to say is that the timing of hippocampal activity in relation to sleep spindles is also phase specific. So just like I showed you with theta, in terms of sleep spindles, there's also a kind of consolidation dependent firing at sleep spindle peaks versus troughs. All right, so the last point I want to say is that Raquel Guthrie will tell us in about an hour whether dissociated states that I just showed you happen in the rat also occur in between human brain areas. And that would have an effect on what we want to amplify and where we want to amplify it in the brain. What this means for cognition and sleep-specific interventions is to be determined, but we really do need to take local sleep features into account and precisely time our interventions in relation to them. So thank you very much. Sweet dreams, everyone. And this is who has supported this information through all these years. So thank you.
Thank you, Gina, for a great talk. Any questions from the audience here?
I have a quick question. I've been a big fan of this Aura ring, which I'm sure you know about.
Oh, yes.
It may be a tiny bit off topic, but I'm wondering if you could comment, in terms of, what we can monitor and maybe, more so, what do we have control over in our lives in terms of improving our sleep beyond getting more sleep?
Yeah, I think that's a fantastic question. Thank you for asking it, because another place which is rapidly developing right now is the technology to be able to detect what sleep state we're in. And what I just showed you in terms of slow waves and spindles means that maybe just detecting what state you're in isn't enough. You need to also detect what is happening in the brain during these various states, because those sleep spindles, the density of them is correlated with our ability to learn through sleep. And their nesting with the slow oscillations is really critically important. So I don't think it's useless at all, but I think that we need to continue developing how we can measure sleep features, not just sleep states. And that's something that we're working on. Karina Koos, I showed you, who's starting the neuroscience IDP this fall, is working on a really great way to detect, an easy way to detect precisely our sleep states and sleep features. So thank you, Anne, for asking. I want one of those rings, by the way. Yes, Nanthia.
We have a couple questions in the chat. One is about naps, whether power naps work, and can you offer some insight?
Yes, I am a big fan of power naps. One of the things that builds up the longer we're awake is adenosine power naps are long enough to help convert that adenosine back to ATP and give us the power to do what we need to do during wakefulness. Also, Sarah Mendick showed that a nap that includes REM sleep, which needs to be longer than a power nap, it needs to be about an hour and a half or almost 2 hours. If it includes REM sleep, then it can be as good as an entire night's sleep in terms of what it does for our cognition, because it contains all of those sleep states that I showed you, a longer nap. So just don't push those naps too close to bedtime, because otherwise you're not going to be able to fall asleep. But nap in the middle of the day when you feel the need, go for it.
We have another question here. Are there other redundant pathways besides locus coeruleus for memory consolidation during sleep?
Redundant pathways? Is that what the question was?
Yeah, are there other redundant pathways besides LC for memory consolidation during sleep?
Oh, well, there's lots of other things that are important to memory consolidation during sleep besides locus coeruleus. And you also need the silence of the locus coeruleus during REM sleep in order to accomplish depotentiation, which is important for cleaning synapses that we don't need anymore. So you need locus coeruleus to be active during slow oscillations, but there's also serotonin. Serotonin is really important, and acetylcholine. Those three neurotransmitters, of course, glutamate. You need glutamate to accomplish LTP as well. So yeah.
And then a question for you and I, in humans, is there a minimum number of scalp electrodes that allows reliable analysis of phase for theta or beta oscillations from the medial temporal lobe?
Oh, wow. Okay. Nanthia, that's a great question, and I will attempt to answer it. But from the medial temporal lobe, in humans, unfortunately, you really need a depth electrode, but you can record vestiges of theta with careful techniques from scalp electrodes. It's unclear to me as yet what phase of theta from a scalp electrode corresponds to phases of theta in depth electrodes, but Nanthia, maybe you can answer that.
Well, I hope we can answer that next year at the symposium. We have a graduate student working on this question with simultaneous scalp EEG and intracranial recordings from the medial temporal lobe. And so, Gina, we should definitely talk more about this project. That's why I love these symposiums. So hopefully we can answer it. But as of now, it's yeah, I agree. It's challenging and to record these signals from the deep brain areas, but perhaps with modified scalp electrodes that cover more of the temporal areas and simultaneous intracranial EEG in patients who have permanent implants, that may be a promising way to look at these things in the future. But great question. This is perfectly relevant for the conference like this today. And then I think we're almost. Oh no, we have another minute. Okay, so we'll take a couple more questions. Is there an optimal length or time for power naps, or would it be possible to reach REM sleep in a shorter nap?
It is possible to reach REM sleep in a shorter nap if you're depressed, because REM sleep comes earlier, and but also if you're REM sleep deprived. So because REM sleep is homeostatically regulated, if you're particularly deprived of REM sleep, you will have REM sleep. You can even have sleep onset REM sleep. People with narcolepsy have sleep onset REM sleep all the time. Babies have a lot of sleep onset REM because their pressure for REM sleep is so high. And so if your pressure for REM sleep is really high, it is quite possible that a shorter nap you can get REM sleep. You certainly will get that N2 stage because that comes early, and that's the sleep spindle stage, also important for memory consolidation. Yeah.
And then we have one last question from Dan. Dan, do you want to ask?
Oh, great. Yeah, thanks, Suthana. Gina, great talk and topic. For those of us who are clinicians, especially surgeons, we go on 30 hours without sleep, and then sometimes make it up over a 10-hour, 12-hour block period in the weekends. How does that work, and is that useful?
So that's a really great question we don't know the answer to. For example, in rats, we are giving them a strong learning task and then having them sleep right away. We give them the learning tasks during the light period when their sleep is high anyway. And that immediate sleep, or the sleep within the first 4 hours after that learning task, is really, really critical. But, and like I said, a nap is as good as a night. So if you give a human something important to learn and then give them a nap, they can consolidate that thing within the nap time. But it appears that sleep at night, just following a day's activity, is probably also sufficient, because the nap gets you as much gain as an entire night's sleep, that if you wait 12 hours to sleep at night. Whether you can delay that beyond that time, I think the answer is no, because if we sleep deprive rats or humans, even one night after they learn something new, they will never consolidate that. They have to relearn it all over again when they're well rested and then consolidate it. So yeah, I would not recommend that if at all possible. Avoid that when you're trying to learn something anyway. Thanks.
All right, thanks, Gina. And we'll circle back to the other questions during our discussion.
And let's move on to our third talk for the technology session. It is by Daniel Aharoni. So he's the assistant professor in the Department of Neurology. And he's gonna speak about optical modality for neural recording. So Daniel, please go ahead.
Thanks, Dejan. Thanks to all the organizers for putting together such a great set of very interesting talks. Yeah, so I'm going to be talking about work that really started at UCLA now 8 years ago to develop open source wearable microscopes to image neural activity in freely behaving animals down to the size of a mouse. And so this started when I started my postdoc across Hemong Golshani, Alcino Silva, and Balti Cox group. And now I have my own lab here at UCLA where we're continuing to develop this technology forward, always in an open source manner, really motivated by getting this technology in as many people's hands as possible. And not just for them to be able to use the technology, but for them to understand and afford the technology. And so we do a lot of developments in my group in close collaboration with a lot of groups at UCLA. So I'll give a little bit of a background of the overall project, and then I'll talk a bit about what we've done over the last year or two. So we call the systems we've developed Miniscopes, and it really is a bunch of hardware, firmware, and software. But on top of that, we spend probably just as much time developing resources to share all the work we do to the neuroscience community. So the base system of sort of all the stuff we develop is some type of head-mounted miniature microscope. For the most part, they're developed to be light enough to be worn on the head of a mouse. So, you know, we aim for in the ballpark of 2 grams, but mice can, after being acclimated to the weight, can carry maybe under 5 grams reasonably comfortably. So we have a head-mounted microscope. We then have some off-board acquisition hardware that's all custom made by us. And then those two things are connected by a thin, flexible cable that provides power, communication, and data. One thing that makes our system somewhat unique compared to other miniature microscopes out in the world is that we can carry all of this power, communication, and data over a thin, flexible, commercially available coax cable. So the standard systems run on about a millimeter diameter coax cable, and those can also go down to about 1/3 of a millimeter in diameter, which is critical for getting natural behavior out of animals. And then we have our open source software that connects to hardware we develop, usually over USB. And then that can integrate with other third-party equipment, behavioral cameras, or other devices you use to control experiments. And as I said, we spend a lot of time making sure all the stuff we develop is accessible and understandable. So this is built around our website, miniscope.org, as well as some wikis through GitHub, as well as all the work we do is posted through GitHub. And this is designed with 2 kind of groups in mind. So the vast majority of people are users, and they just want to be able to get all the components, understand how to assemble them through our online tutorials and videos, and get up and running. But we also know that this technology is really still at its infancy and can really be pushed forward, not just by us, but by a lot of groups. So we also share all of our kind of development in terms of source code and circuit schematics. so that other groups can take what we've done and really build on top of it, which has been really great. And I think, maybe about a dozen different tools have come out building off of our original work. And then we also, I think, since starting this and sharing it from a few years ago, there's something like 500 or 600 labs at least that are using Miniscopes. I think now close to 3,000 microscopes that we've developed have been built across 18 countries. And then we also do in-person, small workshops, larger kind of one-day workshops, and now I guess virtual ones because of COVID. But we really are here to get these tools in the hands of as many people as possible. So what does the system look like? So this is our previous generation miniature microscope. It was about 3 grams mounted on the head of a mouse. It allows for an animal to freely behave, in this case, in a small open field, while we are streaming neural imaging data from them. For the most part, what I'm showing in these slides is imaging GCamp6F in hippocampus, the CA1 layer of hippocampus, unless I say otherwise. But what you get with this is the behavior of the animal, as well as streaming neural data out of the in this case, hippocampus. So in the top right is just the raw data that's being imaged with a small image sensor. And all those little dots that are flashing are cell bodies. And we can do some minimal processing. In this case, it's stabilizing the motion of the brain. So the brain has the consistency of jello and wobbles around by plus or minus 5 to 10 micrometers. So we have to correct for that. And then we can do a quick conversion from just the raw fluorescence to the percentage change in fluorescence. And so that's what we're seeing in the bottom right. And then with some analysis techniques, we can identify individual neurons, extract out their activity, and put that in the context of behavior. And then our systems are an order or two of magnitude more affordable than commercial options. And what I'll show in a bit is I think not only are they by far more affordable, but they're definitely the most advanced and robust systems as well. And then the way we're able to do these types of imaging experiments, we can pretty much target most brain regions. And this oftentimes involves a viral injection of a genetic reporter of neural activity, like GCamp. You can also use transgenic mouse lines as well. In this case, we do an injection in CA1 of hippocampus. We then come back, aspirate the tissue above the brain region we're interested in. If it's cortex, we can just put in a cranial window, which I'll show in a second. And then we implant a grid lens within a few hundred micrometers of the cells that have been labeled that we want to image. And then we cement on an aluminum base plate. And then when we want to image, the Miniscope just snaps into the base plate. You can record your experiment every day across a month or two. And then the Miniscope just gets unmounted between the tools. With all that together, looks something like this. So this is a mouse in the top left running on a 2-meter linear track, recorded from Hemant Golshani's lab. We're imaging monochromatic fluorescence, but after we do our processing, we can identify individual cells, attribute fluorescence to those cells. And so what we're seeing in the bottom left is a pseudo-colored video of the neural activity of, I think in this animal is about 500 active neurons across a 10-minute recording. And then we're showing some of the activity of 50 of those neurons on the right. So every time you see a burst of fluorescence, that's a bit of neural activity that we can then tie in with behavior. And importantly, this technique doesn't just give us the temporal dynamics of the population of cells, but we can also see their spatial distribution. And what that allows us to do is record on day 1, record a week later, record a month later, be able to match up the fields of view across those recordings, and with some simple algorithms, be able to, with a high degree of confidence, say the cell we saw on day 1 is the same cell we see a month later. And now we can do longitudinal studies across populations of hundreds, or even in some systems I'll show in a second, thousands of neurons across weeks or months. So this is a recording across a month. We get really nice expression. We get roughly pretty stable numbers, cell counts across long periods of time. In this case, on the order of 500 cells, and then we can track those individual cells across these sessions. One thing we've started developing a few years ago, this is also openly available now, and we're working on advancing it is making systems that are fully wireless. So we put a lot of time into making the cabling between our miniscopes and acquisition hardware as thin and flexible as possible. But in some cases, you want to get rid of that tethering or cable altogether. So we have these wire-free miniscopes, we call them. They weigh a little bit more because in this case, the mouse carries a battery on the head of the animal, and all the electronics are different to store imaging data on a micro SD card. This is now a mouse running on a little over 7-meter long track without any tethers. We can record for about 25 minutes with similar image quality, extracting hundreds of cells. This allows us to now do experiments in environments that are really just not feasible if there's cabling. It also opens up the ability to record from multiple animals simultaneously without cables getting twisted together. And you can imagine you can have them now go through tunnels and things in this case. I'll skip over this. So we also, in the last year and a half, developed a brand new embodiment of miniature microscopes, which we call the version 4 miniature microscope that's now openly available. And I think maybe 400 have been built over the last year. And some key components of this is the system now incorporates a tunable lens. So through our software, we can just adjust the focus. We're imaging, how deep we're imaging the brain. The system is now quite a bit lighter. So it's a little over 2 grams, about 2 centimeters tall. Because of the optimized optics and image sensor, we can image for a lot longer than we previously could due to photo bleaching. So this is an example, just looking at a resolution test slide of us adjusting the focus through software, which is super convenient. You never have to touch the animal or the microscope on the animal's head, and you can repeatably dial in the same focal plane day after day. We also put a 9 degree of freedom sensor, magnetometer, accelerometer, and gyroscope on the miniature microscope. So now we can stream out head orientation data. So this is just someone in my lab moving around the miniscope, and the little miniscope logo moves with the animal's head. And an example of this on a rat from Tad Blair's lab. This is a rat running on a linear track, and we can stream in real time head orientation. It's pretty cool, even without seeing the body or the position of the animal. If you've worked with mice or rats for a bit, you can see just the behavior of just the orientation of the animal's head and have a pretty good idea of what the rat was doing during this recording. And for other reasons, we can now, these systems can image through cranial windows, which really wasn't possible in previous generation systems and almost all other miniature microscopes, which makes the surgery compatible with other imaging techniques like 2-photon imaging. So if you want to image cortex, you just do a craniotomy, you plant a cranial window, and now you can image cortex, then put the animal under a 2-photon microscope as well and image through the same cranial window. These are examples recorded by Mega Siegel in Alcino Silva's lab, one across the midline and one just in one hemisphere of cortex. This is also using a microscope that has quite a large field of view that's still 2.5 grams. So it's about 1.5 millimeter field of view, which is pretty great. Now we can start achieving above 1,000 active neuron recordings in 3D behaving mice. We can also image deeper by implanting these cylindrical lenses called Brin lenses. This is a recording of medial prefrontal cortex from Ping Zhao from the Golshani lab. This is about 1 millimeter field of view. This is the raw data coming out of the animal's head. And we do a little processing, and there's something like maybe 800 neurons in this freely behaving animal with really nice data. And I mentioned that part of the goal for some of our new developments is kind of push the length. And so with optimized optics and a much more sensitive image sensor, we can, from a few years ago, we were happy if we could do 30-minute recording before we saw significant photo bleaching of GCAMs. We can now go much longer. So this is a 9-hour recording of an animal in an open field. That's just the occupancy map across those 9 hours. And we get very little photo bleaching across those 9 hours. In this case, about a decrease of 9 to 10% in fluorescent brightness over those 9 hours. We can now do a factor 2 or 3 better than that with some of our newer systems. And I think we're now approaching the ability to record indefinitely with chronically implanted miniature microscopes without significant photo aging. So you can imagine now imaging 24/7 across a month or two. I'll skip over this and I'll talk about one of the microscopes we are finishing developing right now, which is what we call large field of view microscope. So this is long-term goal is this is being developed with Payman Golshani and Michelle Basso's lab to do large field of view imaging in freely behaving non-human primates. But as we were developing this, system was small enough to mount on a freely behaving rat as well. So in collaboration with Pat Blair's lab, we've done a lot of great imaging currently, huge field of view in freely behaving rats. And give you an idea of that field of view, this is imaging, in this case, in a head fixed mouse through a cranial window. So this is the field of view is about 3.5 by 2.5 millimeters with single cell resolution across the entire field of view. In comparison, the blue square is our original miniature microscope field of view. This orange square is the largest field of view being done with commercially available microscopes. And then this is the field of view of what I have been showing, which is what we call a virgin core miniscope. And we can do quite a bit bigger. And in this is on a mouse in the Blair lab, they went from recording freely behaving rats with previous generation miniscopes where they would be happy if they got 50 or 80 active neurons that could be detected in a session, to now they have recordings that get close to 1,000 neurons. So this is imaging hippocampus in a freely behaving rat with close to, I think, 800 neurons detected here. And those are just a few of the tools we are developing. We're also developing miniature microscopes that can generate spatial temporal illumination patterns to trigger individual neurons or patterns of neurons optogenetically. In collaboration with Ali Pasha Vaziri's group, we are working on light field miniscopes that can image volumes at video frame rates. And then we're also doing a lot of work incorporating the behavior side of things with imaging. So we can now have these nice neural imaging tools, but these are generally being used in conjunction with behavioral tools. And so with our new software and some other devices we've been working on, we really want to integrate both aspects of behavioral and neural imaging into a single platform that makes it as easy as possible for labs to do these types of experiments. And so most of what I've shown are developments from my group, from Chang, Lang, and Federico, and really close collaboration with Payman, Alcino, and Tad Blair's group. So thank you. I'm happy if there's time to take some questions.
So next up, we have J.P. Langevin. J.P. is the chief of neurosurgery at VA, and he'll be talking to us about responsive neurostimulation for PTSD.
Thanks, Dan. Just to make sure everybody sees you see this.
Yeah, we see it. Looks good.
Yeah, so this project that we have is RNS, or responsive neurostimulation for PTSD. So it's under one of the Brain Initiative grant from the NIH. And just to put everybody on the same page to start, you know, U.S. has the largest PTSD population due to multiple armed conflicts that we've had over a number of years. And basically, it's a condition where, you know, a patient is presented to a life-threatening event, and as a result of that, they are kind of stuck in a fear state, and they constantly have these vivid memories of that life-threatening event, flashbacks essentially, and trauma reminders. And that leads them to avoid any social situation that could remind them of the trauma. And the last cardinal symptoms is hyperarousal, meaning like they have nightmares, sweating, tachycardia related to the trauma and the reminders of the trauma. In the US, about 30% of the combat vets have PTSD. And just to give a ballpark, it's about $6 billion the cost of the US government. There are good treatments, you know, that includes SSRIs on medication as well as psychotherapy. And the psychotherapy is basically trying to expose the patients back to their trauma gradually to engage the internal circuitry so that they can extinguish the fear. And so that is useful for about 70% of patients. But as you can tell on the far end of the curve here, there's about a 30% that fail to respond. And those are the ones that, you know, we are trying to help with this project. When we look at the brain, it seems like the hotspot, you know, the critical area for PTSD is the amygdala. And so there's several lines of evidence for that. So like in the top slide is fMRI. It's actually a combination of multiple studies here, but showing fMRI of patients with PTSD at rest versus when they're exposed to their trauma reminder. So usually they'll place a veteran inside the fMRI and expose them to a imagery or to sounds of war. And so as you can tell, like the amygdala is lighting up on the symptomatic phase of the scan. And at the bottom, they did a similar exposure, but this time with older, you know, PET scan. And in that study, they also correlated the intensity of the metabolic hyperactivity of the amygdala to the CAP score, which is the clinical score of PTSD, basically matching the metabolic activity to the intensity of the symptoms. On the other end of things, when they looked at Vietnam combat vets who also had traumatic brain injury, they could then classify where the injury was inside the brain. And what they found is if the injury is specifically located in the amygdala, none of those patients ever developed PTSD symptoms, you know, really showing a critical role of the amygdala, not only to form PTSD or create PTSD, but also to, you know, be responsible of the symptoms later on in life. So like to have a resurgence of the symptoms. And then when there are some rodent studies as well that delve into detail of the amygdala to look at the architecture to see what could be critical. And they essentially found that at the cellular level, neuronal level, if you look at the basolateral portion of the amygdala, they actually have distinct populations of fear cells and extinction cells. So depending on where those neurons are connected to into the prefrontal cortex, they accomplish the opposite role, like almost like the visual cortex, you know, to look at shapes. And so along the lines of receptive field in the visual cortex, you can imagine that at any point in time, an individual could have neuronal populations that are mostly active on the fear side or on the extension side. So you could have like, you know, two separate conditions, one where you have mostly extension cells that are activated or near threshold of activation, like the top example, or the bottom example where you have more fear, right? So then you can essentially like setting the tone. So if you're like watching a horror movie, for example, at home and it's dark, then you're probably going to be on the bottom schematic where you have more fear cells that are close to threshold. And this is most likely coordinated by the rest of the brain that is connected to the amygdala, since there's broad connections. So, but in particular, the hippocampus that we know has large connections to the basolateral amygdala, as well as the medial prefrontal cortex. So most likely, those areas of the brain are modulating the level or the threshold of activity of those neurons and determining if somebody is geared towards fear or extinction. And so in the state of PTSD, so the patients are stuck into this persistent fear state, so where they lack the plasticity to revert to an extinction states. And so what happens on the patient that we believe happens on the patients who have very severe PTSD is that the psychotherapy is unable to engage the extinction circuitry because the patient's architecture is stuck into this fear state. So any trauma reminder, as benign as it could be, triggers the actual fear response. And so the patient never learns to extinguish fear. So the role of neuromodulation in this case is to reduce the neuronal activity in those neurons that are hyperactive. And so the idea that we had is to place deep brain stimulation electrode into the basolateral amygdala and to chronically stimulate that high frequency stimulation. So we know from our clinical use in movement disorder that when we do that, we essentially reduce the functional activity of those neurons. And so since the majority of those neurons currently that are active in that area are the fear cells, just by sheer volume, we're going to reduce mostly those neuronal activity. And so reestablish, like in the center here, something that's more neutral. And at that point, the psychotherapy might be able to engage the patient into further extinction states so that the patients can finally experience recovery. We did that in a rodent model of PTSD where the rat is exposed to inescapable shocks in the presence of a cue object. So you can see a small tennis ball. And then 7 days later, we re-exposed the rat to that same tennis ball inside their bedding cage. And so we did control group, basically. So like both rats here, the rat A and rat B were both implanted with the DBS electrode. Then they receive inescapable shock in the presence of the ball. And then they never saw the ball again. One of the rats received DBS for 7 days. The other one was hooked up to external generator without receiving the therapy. And then 7 days later, we put the ball back into their bedding cage. And so you can see the difference in behavior. So like the normal wild rodent behavior would be to approach the ball, to explore it, learn, what it is, like the rat A on the left side. But the PTSD rat, if you will, is the one that's consistently pushing bedding on top of the ball, doesn't want to have anything to do with it, so it wants to have it gone, basically. And at the end, most of the time, it's completely buried. So we measured the time that rodents were spending burying the ball. And so we saw so it's a baseline, very little burying. As I said, usually the rats will explore the ball. And then 1 week post-shock, you can see that those rats that received DBS has a significant reduction compared to the control groups. And then we did a crossover 2 weeks later. And what we found is that the DBS therapy, even though it was stopped for the 2nd week, the results were maintained, right? So like potentially the rat has learned to extinguish fear in relation to the cue object. And also importantly, those rats that initially received sham, but then went on to receive DBS responded, which means that once the behavior of PTSD is established, the DBS, when it's initiated late after the disease has started, can still have a clinical benefit. So then we pursue this clinical trial, so here we just represent a veteran. 4th of July is a very hard time for our US veteran because they see the fireworks, but for them, it reminds them of improvised explosive device from overseas and engaging their fear circuitry. And so our hope is that with using DBS of the basolateral amygdala, we can essentially reduce the fear cell activity and then reestablish normal behavior. So we had the initial results on 2 patients so far. So as you can see, we established our response rate at a 30% reduction in the CAP score. So our first patient was a responder. He reported an abrupt reduction of his nightmares, both in terms of intensity and frequency. So that was one of the first things that disappeared after the initiation of the DBS. And this is the long-term data for our 2 subjects with DBS. And so there's a couple of things to learn from this. So first of all, both subjects responded, fortunately. So they've achieved currently about 50 to 60% reduction in their CAPs. And those are patients that, were essentially off meds at this point because they had failed all the available therapy, including psychotherapy. And so that benefit has been maintained over time. But one thing to look is that you can see how there's so many ups and downs, right? So it's not a clean decrease or improvement on the clinical side. And so every time you see the upswing there, that's a very volatile situation where the patient is actually getting worse, and as clinician, we're concerned, that they could have a suicidal behavior or other problems of that nature. So that leads me to the limitations of DBS, right? So like we can't tell, when the patient comes in our clinic after DBS, if we selected the right parameters. So we're going based on our experience on the clinical side with treating other conditions like OCD and treating a movement disorder. But there's nothing really that the patient is going to tell us that's going to make it convincing that we've achieved the right parameters or we achieved the optimal parameters. The other thing is we cannot predict the clinical outcomes. So we don't know if we're on the right track, right? So month after month, the patient comes in and we're measuring that CAP scores essentially asking the patient how bad or how good their last month was, and we're 100% dependent on that. So we don't know if they are heading in the right direction or if next month is going to be a terrible month again. We also don't know when it's time for them to start psychotherapy. So have we engaged the extension network enough that now we can start exposing them to their trauma and then they'll actually get better from that? Or is it still too early? And if we know that they're going to get better, we also don't know the time frame. So is it going to be in a year, 2 years, 3 years? So we're missing information or hard data, like an objective marker, basically. Electrophysiological biomarker could accomplish most of those roles, you know, so like it's a real-time biomarker that could provide us with a solution on multiple fronts. First of all, it could provide us with on-demand stimulation. So as I said, the amygdala is both important for the extension as well as for the fear response. So if we're shutting it down continuously, you know, we're actually acting a little bit against us when the patient gets better. So we would like the therapy to phase itself out when the extension therapy side of things is started to activate itself. So having a biomarker that could act as a trigger for the device, you know, with on-demand stimulation could solve this problem. In addition, we have an immediate feedback for the stimulation, right? So if you have a marker of the symptoms, you can stimulate the patient and then track over time with the device if that biomarker is eliminated with the stimulation. So like the device recognizes an abnormal signal, triggers a stimulation. So then you have a snapshot of that. So is your biomarker disappear or is it still there? And finally, you can track the frequency of those biomarkers over time to see if you're heading in the right direction. So like you can count how many biomarkers or signals you've triggered over the past month. So you have more than just the clinical scores. So as a first step for that, we had our second patient with PTSD, who prior to the placement of the final DBS electrode, we placed the recording depth electrodes from epilepsy. And then the patient underwent the IAPS, which is basically they're presented with images. And some of them are negative, positive, or neutrals. And we asked the patient to rate those images. And we were encouraged with the result on that signal of patients because we noted that after the presentation of negative images, the patient had an increase in the theta frequency range, approximately 500 milliseconds after the presentation of the image. And that turned out to be almost a mirror image of what had been reported previously in the rodent literature when rats are being presented with a fear conditioning task. We are then repeating these results in epilepsy patients who have RNS. So again, that's an example of our IAPS images. So we're doing this with Nanthia Suthana in her lab. And as you can see, so there's just a representative image where approximately 500 milliseconds after a presentation of negative images, you have this increase in the theta range frequency. And so far we have 8 subjects, and it seems that the results are carrying over across those subjects, again with theta power increase about 500 milliseconds. And so this we've reached out to the device maker NeuroPace, and it seems like this is going to be a signal that can be recognized by the device and act as a trigger. We're also looking at other areas of the brain, obviously, to find additional biomarkers or to see if this one is also relevant. And the other thing that we're looking at is the same signal to see if there's a difference between patients with anxiety disorder and patients who are without anxiety disorder. So is there something about the signal that it's stronger, let's say, or different inside patients with anxiety disorder? So it's still early on, and we're still analyzing those results. But we're moving forward with our trial where basically we're going to place the RNS NeuroPace system with bilateral hippocampal amygdala electrodes. And essentially, instead of recording seizures, we're going to be recording that theta signals in the lab with Nanthia Suthana, as well as real time. So we have our psychiatrist who is going to be exposing the patients to their trauma while recording the device live. So I think I'm out of time. So just in conclusion, as I mentioned, so the important clusters of PTSD symptoms appear to be linked to the amygdala activity. And we've shown, at least in 2 patients, very preliminarily, that DBS of the basolateral amygdala can improve on those symptoms, but it's not a perfect therapy. Again, because we can't really track what we're doing over time. It's hard to decide, if we are delivering enough or too little energy, and we can't see if the patient is getting better objectively. So we believe that the next step for us is going to be to use this closed loop DBS solution, and we're using the responsive neurostimulation device from NeuroPace. And just finally, I just wanted to thank all the people that are helping me out on this project, and in particular, Nanthia Suthana, who's obviously critical into providing us with the data on the ePhys side.
Great, thanks, JP. We're a little bit out of time, but we have time for one question if there are any. Okay, thanks, JP. We'll move on and we'll have a discussion at the very end, so stay on.
Thank you.
Okay. All right, so last but not least, we have Ausaf Bari, who is our new division of functional neurosurgery head. He's going to be talking to us about deep brain stimulation of the sublingual cingulate cortex for medically refractory chronic low back pain. So go ahead, Ausaf.
Thanks, Dan. Can you see the slide?
Yeah, looks good.
Okay, I'll try to make this quick since we're running out of time. So I'll be talking about deep brain stimulation of the SCC for chronic low back pain. These are my disclosures. So deep brain stimulation is currently approved for these 5 indications, as you see here. Dr. Markovic kind of touched on movement disorders and epilepsy earlier. It's also approved for OCD and dystonia. But we're moving into a realm now where we're trying to treat these neuropsychiatric disorders like Dr. Langevin was talking about, PTSD, addiction, anorexia, etc. Today, I'll be talking about chronic pain. Eventually, where this field is headed is into enhancement of function, which has its own ethical issues, but we're headed into that realm in the next few decades. So one of the nice things about DBS is that not only is it therapeutic for many things, it's also an opportunity to map the brain. And it's been used extensively to map mood, affect, and pain circuits in the brains of patients that have undergone DBS for various indications. But why should we think about developing therapies for pain in particular? Pain affects at least 23.5 million adults in the US with an enormous cost to society, approximately 635 billion, very high prevalence. In fact, it's the most common reason anyone will see a physician in their lifetimes. And it's led to this increase in long-term opioid use and the opioid epidemic. And the most common subtype of chronic pain is actually chronic low back pain. So neurostimulation for pain is not a new idea. There's been many forms of neurostimulation, invasive neurostimulation, that have been either offered or postulated to control pain. These include motor cortex stimulation, deep brain stimulation, and spinal cord stimulation. So today I'll focus on deep brain stimulation. And this actually gets to this point that Dr. Markovic brought up, which is very important. It's not necessarily the technology that is the bottleneck here. It's really trying to identify the circuits and the relevant location that we need to go to in order to treat pain. And traditionally, a lot of focus has been spent on the spinal cord to treat the nociceptive component of pain that eventually gets up to our consciousness levels. And today I'll make a case for deep brain stimulation. So traditional targets for DBS include the PAG, the periaqueductal gray, and the thalamus. And it's been found that the PAG, PVG stimulation is better for nociceptive pain, whereas the VPM, VPL thalamic stimulation actually is a little bit better for neuropathic pain. If we look at the overall pain reduction, we see that there's that for nociceptive pain, we see about a 61% success in these large conglomerate studies, and about a 42% reduction in neuropathic pain. And if we compare targets, we see that the VPL VPM here again is better for neuropathic pain, whereas the PAG, PVG has been shown to be more effective for nociceptive pain, but there is some overlap between these targets. And there's a lot of variability as well. So this here is a long list, laundry list of different types of pain, including central post-stroke pain. We have here highlighted is the low back and skeletal pain. That you can see if you look at the percent success of DBS, it's highly variable depending on the subtype of pain. And if you focus on just low back pain, we see about an 80% success rate according to this study. So one of the things to keep in mind is a lot of the previous studies were limited in that they were really looking at these heterogeneous populations of patients. And there's a case to be made that there's no one single type of pain. There's obviously many subtypes and they have different responses to different forms of neuromodulation. So DBS for pain has about an overall 50% efficacy given the current targets that we're using. It's more effective for nociceptive pain and better for peripheral versus central pain. However, there's more data that's needed, especially functional neuroimaging and other functional studies to find better targets. And so what I'm going to be talking about is this paradigm shift that we're proposing for neuromodulation for pain, which is to think about this concept of pain versus nociception, whereas nociception is more the sensory discriminatory phenomenon. We think of pain as actually a subjective and a more conscious perception, which lies at this intersection of somatization, affect, and cognition. So really what we're proposing is that we should no longer be thinking of pain as purely sensory. And in particular, chronic low back pain and chronic pain, we should think of more as an affective disorder of the brain. And so really the crux of this is that pain really is in the brain. And especially suffering from pain lies in the brain itself and not in the periphery. And this has been kind of shown in studies by the Abkarian group. As shown here, you have a patient who's got chronic back pain. And if you do functional neuroimaging, you can see that this patient's spontaneous pain related to their chronic low back pain is engaging these frontal limbic circuits. But when you produce acute nociceptive pain in this patient, you get this kind of a pattern that is very similar to thermal pain in normal patients. So here you can see there's a distinct pattern difference between thermal nociceptive pain and chronic low back pain, which we're proposing is more of an affective disorder. And again, I'll skip over this, but this is more neuroimaging pointing in this direction. And this was really from the crux of our proposal that we sent out to the NIH under a UH3 mechanism. Now, this is not something new to propose affective areas for controlling pain. In fact, this is a very old slide. This has been known since the 1950s that you could go in and you can do ablation of this area. So here we have an affective area, which is the anterior cingulate area. And this has been used by neurosurgeons for many decades to treat chronic pain. And you can see here at UCLA, we were doing this technique using MR thermography. So basically making a small burr and ablating this area bilaterally. And this is often a technique that we use now to treat suffering from chronic cancer pain. And interestingly, these patients will tell you that they still feel the nociception. They can still tell you that there's a sensation associated with the pain, but that after the ablation, it no longer is bothersome to them. So you reduce or eliminate the affective component, which is really what is causing all the debilitating symptoms of pain and leading to the social burden. So we propose the state space model of DBS, and we think of pain as lying on these three axes of neuronal activity, where you get combinations of affect, cognition, and physiological activity. And these particular combinations results in this chronic pain state. And the goal of DBS is to push neuronal activity out of this pain state, and with closed loop DBS eventually to keep it in this state that's outside of the chronic pain state. And so I'm not going to go over these two. These are older groups that have targeted different areas. This was the anterior cingulate that was targeted by the Oxford group. Machado et al. Targeted the ventral striatum. Both of these studies had limitations. And in general, the limitations were the inclusion of heterogeneous groups and a disproportionate focus on central and neuropathic pain types. And also this lack of crossover or blinded withdrawal design, which has now been proposed in many areas of DBS trials as a better design. So basically, we were awarded this NIH UH3 grant, which is a single center study in which we're proposing to implant 16 subjects with chronic low back pain in a randomized crossover double-blinded withdrawal design. So basically, there's an optimization phase. So these are patients that come in with chronic low back pain. So we're using a homogeneous population of patients. So these are not central thalamic post-stroke pain patients. And I think this improves the generalizability of the study. In this optimization phase, the patients are implanted using tractography, which I'll get into the most relevant area within the subgenual cingulate cortex. And we find the best parameters for each patient in an open label fashion. Once we find that, we then randomize patients to their best parameters, which is active versus sham stimulation for 3 months. And then there's a crossover. So 3 months on, 3 months off in a double-blinded fashion. And then there's an open label period. In the open label period, what we will do is we propose to look at the tractography and the wiring of these patients. And we're going to identify patients with a fingerprint of good stimulation versus bad stimulation, or effective and non-effective stimulation. And by studying the patients that get beneficial stimulation and looking at their wiring pattern in their subgenual cingulate, we will try to use these directional leads. So we're using directional hardware here with segmented leads that allow us to selectively engage different white matter tracks. We will try to push the non-responders into responders by engaging the relevant tracks based on the responders. And so this is where the subgenual cingulate lies in the brain. It's been used for many years now off-label for depression. And it's an area where if you do glucose metabolism studies in neuroimaging, this is an area that lights up when patients think about sad memories and in patients with depression. And in patients with depression that underwent DBS of this region, you can actually reverse their symptoms and also reverse the glucose utilization in this area. Evie Toslaki in our lab is a postdoc who did some of the seminal work in tractography that showed that this area is highly connected to the ventral striatum and the anterior cingulate region, which formed the crux for our kind of grant, which said that we're going to target this area not only for its local effect here, but really because it has widespread connectivity to these other areas based on tractography that are relevant in the emotional processing of pain. So we have selective bundles here, the forceps minor, the uncinate fasciculus, and the cingulum bundle. And so these are some of the main pathways that we're targeting with this strategy. So this is our hypothesis that this will be mainly, it's a feasibility and preliminary efficacy study. And so we hypothesize that this will be safe and feasible in patients with medically refractory chronic low back pain, and that it will result in a significant reduction in the patient's opioid use in the responder group. And we're using DTI to optimize our response, and response will be contingent upon stimulation of these 3 major fiber tracks, these affective fiber bundles. And we hypothesize that tractography will be used to optimize the response in the non-responders based on the directional targeting capability of this specific device. And so in conclusion, I just want to put a plug out for functional neurosurgery in general. So we have the surgical neuromodulation brain mapping lab that I direct at UCLA. And this is just a slide showing that these patients are awake during our operations. They're able to perform tasks while we have access directly to their brain and to their circuits. So we're able to not only record from the basal ganglia during these operations, but we were able to put in subdural strip electrodes so that we can actually record from areas like the dorsolateral prefrontal cortex and the ventral medial prefrontal cortex. So we can study other areas that I'm interested in, for example, reward, as you can see here. And we combine this with structural neuroimaging in my lab to better understand and map the brain in terms of its affective circuits and emotional brain mapping. So with that, I know that was kind of a whirlwind, but I wanted to keep it on time, and then we can take questions during the question period. These are just my collaborators. Thank you.